Thank you. Hi, everyone. I will be talking on nanoscale segregation of synaptic scaffolding proteins SAP97 that follows first order phase transition. The previous talk of Mini Jo's, she has explained the super resolution microscopy techniques which she has used to understand the molecular heterogeneities inside living cells. So I will be also using similar techniques, which is stochastic optical reconstruction microscopy for my studies. So let me jump onto my research question. And just before that, I will give you a brief introduction on the SAP97 protein. So SAP97 in mammals is also called as in Drosophila as DLG1, and HDLG in humans is a ubiquitously present protein in all in different types of cell types, as well as it is also an evolutionarily conserved protein. It's also predominantly seen in cell-to-cell junctions, especially in neuronal synapses, as well as in immunological synapses, and also in epithelial cell junctions. And also, the expression of SAP97 is observed to have multiple isoforms with respect to the alternative splicing happening at the N-terminus as well as at the C-terminus part. And also, SAP97 is the only known membrane-associated guanylate kinase protein, which is capable of directly interacting with AMPA-type glutamate receptor, which is activity-dependent, which is the GLUA1. And also, the ectopic expression and silencing of the gene is observed to be seen to alter the synaptic firing frequencies. And also SAP97 is also reported to be seen as impaired in multiple types of disorders such as cancers, and also in neurological pathologies such as schizophrenia and bipolar disorder. So SAP97 has different types of protein domains and a multitude of interaction partners. Let me take your focus onto a particular interaction partner, which is a calcium sensor, which is calmodulin, and it interacts with a region called as hook region, which comes at the C-terminus part, and it is also called as I3 region. So the postsynaptic density of a synapse, it is in recent studies, it is considered as a phase-separated compartment with different sort of signaling molecules present. And looking closer to the postsynaptic density region, near to the receptors, there is a primary interaction with a network-like structure found by the scaffolding molecules. And these scaffolding molecules enable these receptors to dock at the postsynaptic density region with the actin cytoskeleton. So SAP97 is one among these scaffolding molecules which has a direct interaction with the AMPA-type receptors. So now recent studies have been shown up that the synapse, as say, is getting stabilized as a membraneless organelle undergoing liquid-liquid phase separation. There are multiple studies coming up, and this is actually a hot topic right now. So let me just go into a fundamental of phase transition. Let's consider two different types of molecules which are separated in a container with a barrier, and it is having, the system is having a low entropy. And when we remove the barrier, they are free to move and they can form into a higher entropy state. So if these molecules are allowed to mix, demix again, and is it possible? And if it is possible, then what drives it? So let's consider the intermolecular interactions. Let's say if the molecules are similar, they have a homotypic interaction. If the molecules are different, we can consider it as a heterotypic interaction. And only when the average homotypic interaction energy is lesser than heterotypic interaction energy, then we have the phase separation happening inside live cell systems. So here, what we are trying to do is combining classical nucleation theory along with super resolution microscopy to understand the cluster formation in SAP97 molecules in cell systems. So here in CNT, the subcritical clusters follows a Boltzmann distribution, and from that we can calculate the Gibbs energy of formation. And here we are assuming that the clusters are in spherical shape, and also the distribution of molecules inside the cluster is uniform. And from here in delta G, we get two components, which are the surface energy component and also a bulk energy component of the cluster. And from here, we move on to the phase separation, that is which we can see in neuro 2S cells using super resolution or single molecule fluorescence intensity. And in neuro 2S cells, we saw that SAP97 clusters followed a first order phase transition. So coming to more questions, like in Neuro2 cells, what we saw was endogenous SAP97. So we need to know which isoforms of SAP97 is present there, and also what is happening inside the neurons, where and or what isoforms are present in neurons. So we did a mRNA level study to understand the expression level of SAP97 in postnatal rodent brain and also in neuroblastoma cells, and taking your attention to the C-terminus splice variants, which are I3 and I2, and I3, which is capable of interaction with the calmodulin, was predominantly absent in Neuro2 cells, while in hippocampal cells, we could see that there was a more or less equal expression of both I3 and I2 variants. So this suggests that Neuro2 cells is a predominantly I2 environment, and coming to the neurons, we try to look at different developmental time points at an earlier time point, which is 7 days in vitro cultures, and also at a later time point, which is 14 days in vitro cultures, which are mature neurons. So we could see that at 7 days in vitro, we could see the smaller cluster formation of SAP97, while when it was matured, the SAP97 clusters were growing larger. Activity-dependent conformational changes of SAP97 was already studied, and with activity, the calcium entry will be enhanced, and the calmodulin is able to bind to the I3 region of SAP97. This was shown by Parmani et al. And also in Lin and Jeffers et al. They showed that based on the interaction, the C-terminus part of SAP97 can interact to the N-terminus part and form a closed conformation. And when this interaction is perturbed, the SAP97 can form an extended conformation, and these different conformations themselves can interact with different interaction partners per se in neurons, the glutamate type receptors, which are AMPA type and NMDA type. So this shows that there is an isoform specific interaction difference in SAP97, and we try to look at what is the effect of phase separation when there is a calcium level perturbation happening. So we tried a pharmacological treatment, one with topsygargin to increase the intracellular calcium level, then with W7, which is an antagonistic inhibitor of calmodulin, and bacta-AM to chelate the intracellular calcium. So the effect was seen in Neuro2 cells as when there was an increase in calcium level, or when the calmodulin was reduced or the intracellular calcium was depleted, we could see that whether perturbation increased the formation of clusters, SAP97 clusters inside Neuro2 cells. So this, we need to keep in mind that this is an I2 alone in isoform environment, and here whenever we were perturbing, there was a increase of recruitment of molecules into the clusters. We looked at the neurons. In earlier neurons, there was no effect with calcium perturbation, and at matured neurons, we could see that when the calmodulin was inhibited, there was more recruitment of molecules. There could be more recruitment of molecules, and while when the intracellular calcium was chelated, the critical cluster radius was reducing, so suggesting that lesser number of molecules are being recruited towards the SAP97 clusters. So this shows that there is a time-dependent change as well as an isoform-dependent variation in SAP97, both in Neuro2 cells as well as in neurons. So we have observed that SAP97 clusters are differentially regulated with altered calcium levels as well as the isoform composition. So we need to check what is happening actually to the cluster parameters. For that, we did a rank order analysis. Here we take the images, we do the data extraction, we rank order them, and then we plot them for a simple linear regression. With the simple linear regression with control and treated data set, we have a slope of the plot, and with the slope, whether it is shifting upwards or downwards, we know that whether there is a shift, whether it is upscaling or downscaling, and we take that slope as an arbitrary scaling factor, and then we arrive at an actual scaling factor. So here you are seeing the average intensity of molecules, and here you can see that the simple linear regression, and from here we are taking the arbitrary scaling factor, and we take 100 different scaling factors within the range of this arbitrary scaling factor, and we scale down the treated data set to match up to the control data set with these 100 scaling factors, and we plot the p-values for those control and scale treated data set with KS test. And these p-values are plotted here, and the scaling factor showing the highest p-value is taken as the actual scaling factor. And in the cumulative distribution, we have shown that the scale treated data set and the control data set is falling in line. So this suggests that the scaling factor which we arrived at is actually the actual scaling factor that is happening inside the change that is happening here. So the cluster parameters were altered in Neuro2 cells, and we could see that both the area as well as average intensity got drastically increased. And this we tried to replicate in neurons, in matured neurons, since there was no change in the younger neurons, and we could see that similar results which we saw in phase transition. We could see that the cluster parameters were changing here also, and we could see that when the calmodulin with W7, with the calmodulin was depleted, there was more recruitment of molecules without a change in the area of the clusters, while when there was depletion of intracellular calcium, we could see that the recruitment of molecules were reduced while the area of the clusters were not perturbed. So concluding, the N2S cells showed that there is a differential recruitment. When there is a calcium perturbation happening, there is more recruitment of molecules inside the clusters, and also when the calcium levels were perturbed, and also coming to the neurons, we could see that when the calmodulin was blocked, we could see more recruitment of molecules, and when the intracellular calcium was reduced, we could see a diffused or the retraction of molecules from the clusters to the diffused medium. And also, we also did FRAP studies to understand the mobility of the molecules inside the cells with variable calcium levels, and the data is not shown. And also, further, we are interested in looking at in vitro imaging of these proteins in different in the presence of calmodulin as well as at different calcium levels, to understand the phase separation of SAP97 outside living systems. So altogether, our study is a humble attempt to understand these molecular intricacies of these proteins, which are having high variability in the presence of multiple isoforms, and to extend these implications into health and medicine. So I thank Dr. Deepak and Dr. Mini Jose and my colleagues for helping me in doing this work, and CNS administration and all staff, and CBS imaging facility, IISc animal facility for providing the animals, IISc MHRD and UGC for funding the research. Thank you.
Hello. Thank you, Prem, for that excellent talk. We have time for 2 questions. Is there a question? So if not, I'm going to ask a question. So if I'm not mistaken, you're assuming that the phase separated droplets are spherical and they are kind of uniform inside there. But when we look at these clusters that are forming in the neuron, they are pretty much like, there are probably 7 or 8 proteins, right? Maybe 10, 20 in the very large clusters. So can you really use a spherical approximation and the approximation that they are uniform inside the droplet? Have you done any kind of particle tracking to verify this claim?
Yeah, that's actually a good question, but we haven't looked at the particle tracking per se. So we have just done FRAP studies. So FRAP studies just understand the basic mobility changes that is happening to the proteins, but the particle tracking studies which you are talking about, which is the photoactivated localization microscopy studies, we haven't reached there yet.
Is there any other question?
I missed it. How did you conclude the transition is first order? You showed some graphs and it went too fast.
Yeah, sorry.
Phase transition that you said it's a first order phase transition.
Yes, yes.
How did you conclude that?
Conclude in the sense?
What experimental results?
So that is stochastic optical reconstruction microscopy. So there we are actually looking at the single molecule localization. So with the single molecule data, we are actually calculating the number of molecules inside the cluster, and with that, the number of molecules as the parameter, we are actually deriving the equation. So in the equation, as you have seen, and the delta G is actually an raised to 2 by 3 plus or minus bn. So in that, the number of molecules inside the cluster is taken as a parameter. So here we assume the number of molecules inside the cluster is uniformly distributed. But in actual case, in actuality, this is not very close to the reality point, where there are nano domains which are present inside these clusters. That part we haven't reached yet, so that needs like further data, variables need to be included into the equations. Yes, sure. Yeah, sure.
Is there any other questions? If not, please thank Prem, and Ayan is the next speaker.